The next type of cell division that we find is the cell cycle. Now, the cell cycle, again, is one of the major types of cell division in eukaryotic organisms. So here's our overview of the cell cycle. Now, remember what the cell cycle does overall. It takes one parent cell, and that parent cell divides to produce two identical daughter cells, identical to each other and the parent. The cell cycle can be largely split up into two phases. We have first interphase. So if we say that this is the original parent cell, it's going to begin the cell cycle starting here and going clockwise. So we have interphase first, and then after interphase, we move into the mitotic or M phase. Now, interphase, even though interphase takes the longest, it can take up to 90% of the cell cycle, there's no actual division that occurs during interphase. Instead, interphase is the groundwork. It's the preparation to divide. Now, preparing to divide is very difficult. It's very long. That takes the most amount of work, but there is no actual division that occurs in interphase. So the prep for division, interphase, gets split up into three stages: G1, S, and G2. Technically, the G stand for gap. So we have the first gap phase right here and the second gap phase there. But we can think of the G's as standing for growth and getting ready, and that will help you remember what occurs. In G1, the cell grows, it physically enlarges, and it gets ready or prepares for the next stage, which is S. Now, S stands for synthesis, and synthesis means to make. What happens in the S phase is that we make DNA. So the process of DNA replication occurs in the S phase, where we make an identical copy of every chromosome, every piece of DNA in the cell. After we do that, we move into G2, the second gap phase, and in G2, we get more growth and getting ready for the mitotic phase, what comes next. And after G2 is finished, interphase is done, and we move into the mitotic phase. Now, in the mitotic phase, this is the actual division part of the cell cycle. There are two different things we're dividing here. We're dividing our DNA, and then the rest of the cell divides after that. So division of the DNA is known as mitosis. All right, mitosis is often called nuclear division because we're dividing the DNA that used to be in the nucleus. After we do our nuclear division, or dividing up of the DNA in mitosis, then after that we move into cytokinesis. Cytokinesis can be thought of as cytoplasmic division because the rest of the cell, the cytoplasm, is what divides, and this is where the cell splits into two daughter cells, cytokinesis. So once that cell runs through the cell cycle and divides, we get two identical daughter cells, these two right here. Now, depending on the kinds of signals that these cells receive, maybe both of them will each divide again in the cell cycle. Maybe only one of them will. Maybe neither of them will divide again. Maybe those cells will just differentiate or specialize and become a skin cell or whatever kind of cell they're fated to be. So if a cell is not in the actual process of the cell cycle, not in the process of dividing, then we say the cells are in G0. Okay, that's sort of the stage that they're in. They're in G0 when the cells are not dividing. So interphase, I've told you everything you need to know. Let's talk about the mitotic or M phase in more detail now. So I already mentioned in mitosis that is the where we divide up the DNA that we copied. So we can call that nuclear division. An older term for mitosis is karyokinesis. Karyokinesis, the movement of the nucleus, or what's in the DNA. Karyokinesis, since we're moving DNA around. Cytokinesis is really just cytoplasmic division, again, where the rest of the cell divides. So let's start off with mitosis. Okay. Mitosis gets divided up into 4 stages. We have prophase, metaphase, anaphase, and telophase. Those are the 4 stages of mitosis. It's all just different parts of dividing up our DNA: prophase, metaphase, anaphase, and telophase. So let's begin by looking at what the cell would look like at the end of interphase. All right, end of interphase. The DNA is still in the chromatin form. It's still locked away in the nucleus. All right, so this is what a cell would look like in G2 of interphase. You can't make out individual pieces of DNA because it's that long, stringy chromatin stuff. but it's G2. So we've already made a copy of every piece of DNA inside. Okay. Notice that this is an animal cell because I see these called centrosomes with centrioles on the inside, and I do not see a cell wall. Now, once the cell leaves G2 of interphase, then we move into the first stage of mitosis called prophase. Pro means first or before. So the first stage of mitosis is prophase. By the end of prophase, this is what the cell will look like. So I want you to take a look at this and then compare it to what it looked like in G2. All right. Now, by comparing these two pictures, this will help us figure out what happens during the course of prophase. So one thing we notice is that we have formed the mitotic spindle. Okay, all we had were by G2 anyway. We had two centrosomes. We used to have one centrosome, but that centrosome got duplicated in interphase. So those two centrosomes begin to separate from each other and move to opposite poles of the cell. And as they're moving to opposite poles or ends of the cell, they're also forming their microtubules. And so eventually, by the end of prophase, we have fully formed the mitotic spindle. We have condensed the chromatin into chromosomes. So now we can, under the microscope, we can actually see the chromosomes by the end of prophase. We have broken down the nuclear envelope. So the nucleus breaks down. We have to break down the nucleus because our DNA is locked away in the nucleus. So we have to break it down in order to just be able to access the chromosomes. These are the things that happen during prophase. We form the spindle. The chromatin condenses into chromosomes, and the nucleus breaks down. After prophase, we move to metaphase. When you hear metaphase, think middle. In metaphase, what will happen is that the spindle aligns the chromosomes at the center of the cell. So comparing this picture here to this picture here, this is what happens in metaphase. The chromosomes are aligned by the spindle at the center of the cell. Now, since we call the opposite ends of the cell where the centrosomes are at the poles, then one name for the center of the cell is the equator. An alternate name for the center of the cell is the metaphase plate. And this is where the chromosomes are aligned in the center. After metaphase, we move to anaphase. So comparing the two pictures, the one of metaphase and the one of anaphase here, we can see that what happens in anaphase is that we separate sister chromatids. Sister chromatids are separated from each other. So looking over here, at this point, this is one chromosome made of two identical sister chromatids. But when anaphase occurs, this chromatid up here gets pulled to this pole by that centrosome, and its identical sister chromatid is being pulled towards the opposite direction, the opposite pole, by the other centrosome. And so we see that same chromosome right here. Right? So this is what happens in anaphase. We separate sister chromatids, and each copy gets pulled to an opposite pole or end of the cell, here and here. Now at this point, every chromatid becomes its own chromosome. Over here, every chromosome was made of two chromatids. But now, every chromosome is only made of one chromatid. Okay, once we physically separate the two chromatids from each other, each becomes its own chromosome. So in this picture, I see 1, 2, 3, 4, 5, 6 chromosomes and 12 chromatids. But in anaphase, I see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. We now have 12 chromosomes, and we still have 12 chromatids, but the number just jumped up from 6 to 12 chromosomes. Other thing that happens in anaphase is the cell elongates. It starts stretching out. Now, telophase, comparing anaphase to telophase. What happens in telophase is the DNA, the chromosomes, they have reached their destination by the end. So we can undo all those changes we made in prophase. That's going to mean that we break down the spindle in telophase. That means that we're decondensing chromosomes back into chromatin, and we are reforming nuclei. Instead of one nucleus, there will be two, because this will split into two daughter cells. But these are the things that we do in telophase. We reverse the changes we made in prophase. Breaking down, so we break the spindle down, we reform nuclei, and the chromosomes decondense back into chromatin. Okay? Now, so telophase is the last stage of mitosis, and after mitosis comes cytokinesis. There's a bit of an overlap between telophase and cytokinesis, and that's why you see the two kind of drawn together. Telophase begins, but before it finishes, cytokinesis begins. So there's an overlap between the end of telophase and the beginning of cytokinesis. Now, up to this point, the cell cycle is pretty much the same in plant cells and animal cells in terms of what we do to our chromosomes, how we move them around. That's basically, they're all moved around the same way for plants and animals. However, cytokinesis is a very different process. The reason it's so different between plant and animal cells is because animal cells have a cell wall and plant cells don't. So looking at cytokinesis in animal cells, it's pretty simple. The animal cell just pinches itself in half. Now, the way that the animal cell pinches itself in half is it has if you can imagine, a belt of cytoskeleton called microfilaments. So microfilaments are another type of cytoskeleton, different from microtubules. We get a little belt of microfilaments that goes around the inner circumference of the cell. And that belt of microfilaments begins to constrict or tighten, causing the cell to pinch in half. While the cell is being pinched in half, the little waist or the dip, that is called the cleavage furrow. And so the cleavage furrow gets deeper and deeper until it completely pinches the cell in half during cytokinesis. That's for animal cells. Now, plant cells have cell walls, so they can't simply pinch in half like the animal cell did. Instead, the way that plant cells divide during cytokinesis is they produce many vesicles that contain the components to build a new cell wall. So they start producing all these vesicles, and those vesicles get shipped to the center of the cell where they merge together. So all those vesicles merge together into like one big blob of membrane. In effect, what we've done is we've created a membrane down the middle of the cell. And so we'll have the membrane of what will be one daughter cell here, and the membrane of what will be another daughter cell right there. But we also need to build cell wall, right? Plant cells have cell walls. So some of those vesicles there, they have the components, the raw material to build cell wall. So when those vesicles come to the center of the cell, they merge and they deliver the material that they're carrying, and the cell will use the material dropped off by these vesicles to build new cell walls down the center of the cell, effectively splitting that one cell into two identical daughter cells. While those cell walls are being built, while they're under construction, we refer to it as the cell plate. But eventually that cell plate becomes the new cell walls and membrane of the two daughter cells. I'm not going to play this video for you in my voiceover of the Prezi. If you want to watch this video, which I suggest you do, it's a very good video of the cell cycle, I would open the original Prezi from the other section in the website.